Welcome to my presentation on viscosity-reducing excipients for protein therapeutics. My name is Tobias Rosenkrantz. I'm the head of the biomolecule formulation R&D team in our headquarters in Darmstadt, and I will today talk to you about the following aspects of protein viscosity. First of all, I will describe the challenges that are arising from a high protein viscosity, and I will introduce to you our approach on how to manage protein viscosity. We use excipients for that, and when you use them in combination with each other, you can generate benefits for protein stability, and I'll touch upon this topic as well. And at the end, I will give you an outlook on our technology launch during 2020. How is protein viscosity a challenge? Many MAPs are currently administered intravenously, and this makes dosing fairly simple as there are no volume limitations. So as long as the patient remains in a hospital bed, any volume can be administered to him. A subcutaneous injection offers the benefit that it can be administered by a local doctor in a walk-in clinic and doesn't require hospitalization, so it's much more convenient for the patient. Yet it requires that the proteins are administered at a very high concentration of 100 mg per ml or more, since the injection volume is restricted to between 1 and 2 milliliters per dose. At these high concentrations, as it's illustrated in the cartoon in the pink box, a protein can become viscous, and it can be very difficult, if at all possible, to administer it. And what we are working on is a scientifically driven approach with best-in-class excipients to reduce protein viscosity to the desired level and then enable you to offer an easier administration and a higher convenience to the patient. Why is viscosity such an important driving force of the injection force? And this can be analyzed by looking at that equation. I go through it from the right to the left. The piston friction is a constant that is mitigated by the engineers designing new syringes. The radius of the syringe barrel and the inner radius of the needle, they can be optimized to reduce that force. However, this term is restricted by the fact that a syringe needs to be operated by the human hand, and it needs to one needs to be able to do that single-handedly. The flow rate or injection rate Q is highly dependent on the person that is administering the drug and can therefore not be optimized. And the needle length cannot be infinitely small because the needle needs to be just long enough to reach the subcutaneous tissue where the drug is desired to be administered into, which leaves the viscosity as a driving factor, a major driving factor of the injection force. When do proteins become viscous? In this plot, we see on the x-axis protein concentration, the y-axis viscosity, and the two red lines give the limit of administration, and depending on whom you choose to cite and whom one talks to, these limits can be a little different. That's why I put both of them here on the slide. And we see that at higher concentrations above 200 mg per ml, a lot of proteins become viscous, and this is largely due to molecular crowding. The next neighbor distance of the proteins in solution becomes so narrow that they are interacting with each other, whether there is a specific affinity or not, just like it used to be in a crowded football stadium. At a lower protein concentration, just at, in some cases, 100 and 120 milligrams per milliliter, proteins interact with each other, and this is because they have a high affinity to do so. So we have two distinct concentration regimes: interaction by proteins due to molecular crowding at high concentrations, and interactions of proteins due to a high self-affinity at these lower concentrations. Having established that, the viscosity at high concentrations is inevitable because these interactions will take place, but it still can be mitigated via excipients, and even higher concentrations can be reached, while the viscosity at low concentrations caused by these specific protein-protein interactions is another is another story. These protein-protein interactions are specific. In a sense, these are hydrophobic interactions. These can be charge-based interactions, but they're transient, so they bind and unbind all the time. And what is very important is that these interactions are identical to the intramolecular interactions that stabilize proteins. However, these protein-protein interactions are much weaker than the forces that stabilize a protein because they're much fewer in number. And when one looks at an antibody like it is depicted on the left, there are two distinct regions where the residues causing these interactions and ultimately viscosity can be located. And we find in many, authors have published that in many cases it can be located in the Fab fragment. Summarizing this, if a protein becomes viscous at a lower concentrations, there is a threefold conundrum, because the region that causes the viscosity can be the same region that is responsible for the drug's efficacy, and the interactions are the same that are responsible for protein stability. So the balance here would be efficacy versus viscosity versus stability. And in particular here, it's, in our opinion, very important to use excipients and very efficient to use excipients, because one can reduce viscosity, one can maintain stability without having to change anything on the protein, which would be a risk to the drug's efficacy ultimately. How do we do that? There are numerous excipients for viscosity reduction that are already known and used in marketed formulations, like certain amino acids, but they're not always working sufficiently well, and for some routes of administration, they are not acceptable. Therefore, we developed a portfolio of different excipients that can be used in combinations and that we recommend to use in combinations because, as we will see, there are synergistic viscosity reduction effects that can be observed. And when used together, these excipients offer an improved and a higher stability than when you use some of them by themselves. Where do we start? We're looking at two antibodies in this presentation, MAPC at 150 mg per ml, and assuming a 27 gauge needle, this would require an injection force of 90 Newton if we don't change anything on the marketed formulation. MAPD, 190 mg per ml, 27 gauge needle, we would have 140 Newtons of injection force if nothing is changed in the marketed formulation. First, we look at the performance of single excipients. And as you can see here on the left, this is MAPC in orange, marketed formulation with an increased concentration. The viscosity would be about 100 millipascal per second. And if you use benchmark excipient, excipient 4 and excipient 5, the viscosity would reduce when you use 75 millimolar, and the viscosity would reduce further if you use 150 millimolar. The viscosity reduction wouldn't necessarily double, but the viscosity is going down. For excipient 1, the viscosity is actually increasing when you use that excipient. For MAPD, on the right side, already sodium chloride yields to a viscosity reduction. We are using sodium chloride here because the marketed formulation doesn't contain any salt, and therefore we want to be able to tell the difference of a viscosity reduction by charge or a viscosity reduction by charge plus. And we see also here that there is a viscosity reduction that can be observed, but it's not necessarily directly proportional to the excipient concentration. Now we look at MAPC and we apply combinations of excipient. In red, we see 75 millimolar of the more cationic excipient. In blue, we see the effect of 75 millimolar of a more anionic excipient. And in yellow, we see the calculated viscosity if the effect of the red, the cation, and the blue, the anion, were additive. This is a calculated data point. And in pink, we see the actual experiment. And when the pink bar is lower than the yellow bar, we see synergistic protein. We see synergistic viscosity reduction. A good example here is the combination benchmark excipient 4. The benchmark reduces protein viscosity a little bit. Excipient 4 reduces it further than the benchmark. Together, they should yield to an improved viscosity reduction that is shown in yellow. And when we do the real experiment, then the viscosity gets reduced even further than expected. So benchmark and excipient 4 act synergistically, just like excipient 1 and 4 and excipient 1 and 5, while benchmark and excipient 5 have a pure additive behavior. We can see this similarly to also for MAPD. The viscosity can be reduced by sodium chloride. And when we look at excipient 1 and excipient 4, we see that excipient 1, the more cationic one, would actually be not as efficient as even sodium chloride. It's higher than that. Excipient 4 reduces viscosity. So in sum, the expected viscosity would be a little bit higher than the one of excipient 4 alone. And in fact, we see that together with excipient 4, excipient 1 gives a viscosity reduction, which by itself, when used by itself, has no effect beyond charge. But together with excipient 4, we see that the viscosity can come down. The combination benchmark and excipient 4 would yield to an almost negative viscosity. But since it's a highly concentrated protein solutions, the protein requires space in that solution. The viscosity cannot become 0, but it can become really, really small. And I think here we're really approaching this lower limit of viscosity. So we bring down these injection forces that are required from 90 Newton to 35 Newton for MAPC, and from 140 Newton we can reduce it down to 18 Newton. That is not all. We can also use the excipient ratios to further optimize the viscosity reduction. In this case, we've done this using KD data. The x-axis shows the protein concentration, the y-axis the normalized diffusion coefficient. A negative slope indicates protein attraction, a positive slope repulsion, and the ideal case as far as viscosity reduction is concerned would be a flat line. And here we see in black the marketed formulation. There is a certain attraction in between the proteins. Sodium chloride can mitigate that, and then we can look at the different ratios, and we find that for excipient 1 and excipient 4, a ratio of 2:1 is beneficial. And by optimizing the ratios of the two excipients towards each other, you can further push the viscosity down, and this can give you the extra percent that you need to cross the injectability limit and come out at the right side of it. Why so many excipients? We have tested a protein also at different pHs. So this protein is MAPD, and it's formulated at pH 5. We also formulated it at pH 7.2, and we see that the different excipients have a different viscosity reduction performance, and that the viscosity of the protein in the two different buffers is also slightly different. And then we have done the math and calculated the chemical properties of the excipients using computational chemistry, and we find that there is actually no big change in protonation state. Therefore, the other chemical properties don't really change much. So here you really have a protein that becomes viscous under different formulation conditions for different reasons, and that's why it can be beneficial to have multiple excipients to have a more tailored approach to that. How about the impact on protein stability? We've performed a force degradation study that for us as excipient developer is a little bit different than the way you would sometimes do them. So what we do is we use MAPC, and that is marketed as a lyophilized product, which means when we bring it into a solution, it has a certain inherent instability, and that is good for us because we can see if the addition of excipients improves something or makes something worse. While when you have a fully formulated, very well developed liquid formulation, most of the time we don't see an effect. That's why we're turning to lyophilized proteins and bring them in solution to take advantage of the higher sensitivity of the protein. We use 150 millimolar of the single excipients because this is the concentration you need to be most efficient. And we use 75 millimolar cation and 75 millimolar anionic excipient because in these combinations, we were able to beat the single excipients or get similar results in many cases. The conditions, 80 mg per mil of the protein, the marketed formulations, and we spike the excipients inside, 40 degrees, 75% relative humidity for 28 days. And what we observe is that there is a loss in monomer content in the marketed formulation. And when we look at the combinations of excipients, there is not a big difference towards the marketed formulation alone. However, if we turn to the single excipients, the benchmark excipient and excipient 1, they don't have a big effect on the monomer content beyond what is happening in the marketed formulation. However, excipient 4 and excipient 5 have a very strong negative effect when they are used at high concentrations by themselves. And this brings together the two parts of our technology, the viscosity reduction, a synergistic viscosity reduction, enabling the use of less excipient in a more optimized way, and therefore one can develop a low viscous, stable protein drug that can be conveniently administered to the patient. Last but not least, I would like to briefly talk about the technology launch that is upcoming. If you are interested in pre-testing our technology, please contact us and we can talk about an MTA, and you can test the material free of charge before we launch. And later in the year, we will have a sample kit available without an MTA for you to use. And with this, I would like to summarize. Protein viscosity is a challenge for some routes of administration. An excipient platform can specifically target these challenges, and it's using excipient combinations and shows a synergistic viscosity reduction. And because of that, we're able to better balance protein viscosity versus protein stability, because the synergistic viscosity reductions allows for the use of less excipient, and we have better stability therefore. And what I would like to mention also is that a reduced viscosity can, of course, also yield to an improved process efficiency. And with this, I would like to thank you very much for your attention. And I'm looking forward to your questions. Thank you.